This section of the pituitary gland has been stained with the traditional hematoxylin and eosin stain and is different to that previous section I've just shown you in two very significant ways. Firstly, look at the pars distalis, and you can appreciate that hematoxylin and eosin does not clearly distinguish acidophils from basophils. Both types of cells look orange, even though there are some areas of a bluish stain. So really, the terms acidophil and basophil that I've used here are really misnomers. The granules of both cell types are actually acidophilic. The stain that differentiates them is the periodic acid-shift reagent stain, which is a major component of the polychrome stain that was used in the previous section. Pituitary hormones are glycoproteins and simple polypeptides. The periodic acid shift reagent stains the hormones that are glycoxylated and not those that are simple peptides without sugar moieties. Since this leads to some cells ending up purplish while others are orangish, the conventional appellations of basophil and acidophil have really been inappropriately extended to them. Just remember that collectively the two together are called chromophils. And remember also that the chromophobes continue to stain pale. Secondly, another major difference between the traditional hematoxylin and eosin stain we see here and the previous stain I used before is seen when you view the pars nervosa. The herring bodies are really nicely preserved in this preparation. They appear as oval-shaped packets of yellowish granules. Each packet represents the granule-filled end of an axon sitting next to a capillary, which may not be seen in this section as often the capillaries will collapse during tissue preparation. Herring bodies tend to be clustered together. What you might like to do as a useful learning exercise is to recall what hormone or hormones these granules may contain. As I pointed out when showing you the pituitary stalk, most of the nuclei you see are pituocytes, particularly these nice rounded ones. But obviously, some will be the endothelial cells lining blood capillaries, and they will tend to be more flattened or elongated.